I will talk about the FAIRyMAGs project. So MAGs stand for Metagenomic-Assembled Genomes, and the idea is to build genomes from metagenomic data from microbiome samples. And MAGs are important because the majority of the microorganisms' life is difficult or impossible to cultivate in the labs. But building MAGs is complicated. We have no single state-of-the-art workflows. These workflows can be difficult to design or execute because they require a lot of expert bioinformatics. They require large computational resources, and the resources depends on the taxonomic compositions of the sample. So in the FAIRyMAGs project, can you change the type, please? The idea is to build functional galaxy workflow. That's what we have done. We have built 5 different workflows and a big workflow that combine all of them. And it's really dedicated MAGs building and quality control workflow that you can see in the small location there with many different tools. Next slide, please. One task we did is we really benchmark these workflows together with other existing workflows, like the one from NF-Core, the one from Magneto, and we did that in collaboration with the workflow developers. Next slide, please. We also evaluated our FAIRyMAGs workflows with 4 different use cases from bee microbiome, atmosphere microbiome, algae microbiome, and termite microbiome. And we could really reconstruct really good quality MAGs for each of these use cases. Next slide. One of the next things we did for the FAIRyMAGs was to really develop extensive training materials. We developed at least 2 new tutorials. We updated existing tutorials that were available on the Galaxy Training Network. We recorded these workflows, and we are currently doing the captioning of these workflows so that we can offer them for the next online massive training organized by the Galaxy community, the Galaxy Training Academy that will happen in May. And we offer a new learning pathway to go step by step through that. Next slide. We also designed a machine learning tool to predict a memory requirement for metagenomic assembly together with the Magnify people, and we could identify which models is the best to predict the resources needed for that, and that combine camera knowledge, file size, etc. Next slide. We organized in October a hackathon that were held in Freiburg, but also with an Australian outpost. And in total, we were 23 people and also some people online. We are currently writing a manuscript that will be submitted in bioRxiv in May. And since today, we know that we have 1 talk and 1 poster accepted for the Galaxy Community Conference that will happen in June in Clermont-Ferrand. Next slide. And these FAIRyMAGs workflows, they are available in the microbiology Galaxy Lab that we have been developing also. And they are also available and used by the French national multi-omic platform, AbroMix, that is used for antibiotic resistance and research. And I'm done.